So, oh, sorry, share screen. So, when we finished last time, we were looking at adult stem cells and also induced pluripotent stem cells. And one of the important things we can do with both of these type of cells are create things called organoids. So what is the plan for this lecture? Well, we're going to do a little bit of revision. Then we're going to think about what organoids actually are and in general terms how they're made. We're going to look at particular organoids that model the brain, the stomach and the gut. And we're going to look at some of the possible therapeutic uses. So how these can be used in medicine and to help treat disease. So just a little bit of revision. We know about these Yamanaka factors and how they were discovered, adding these known ES cell factors carefully by removing them one at a time out of 24 and then working out what the minimal number of these that were required. And these were the Yamanaka factors. You remember that they're c-Myc, KLF4, SOX2 and OCT3/4, and the processes by which these were determined experimentally. So I'm not going to go into huge detail there. Suffice to say that these iPS cells, these induced pluripotent stem cells, can be used to make organoids. And hence this idea of the rise of the organoids. This paper in Nature is now nearly 7 years old, and this tells you how long organoids have been being used for and how effective they are beginning to be in medicine. So let's look at the brain first. And what we can do is we can actually grow part of the brain called the cerebral cortex in culture. So you can take your in this case mouse embryonic stem cells and dissociate them, and then we can aggregate them to form embryonic bodies. These grow in culture, and then you can switch the culture conditions in day 7, and these culture conditions select for the formation of the brain. And in these images we can see markers of different parts of the brain, BF1 and EMX1. EMX1 marks the cortex, which is something we're particularly interested in. And you can see that these embryonic bodies are developing the characteristics of brain tissue. And they also form the correct kind of structure. So we have an apical surface of these cortex organoids, and we can see that these cells are dividing in the right area next to the apical surface. So not only do we form cells that are similar to brain, we also form structures that are similar to the brain. And this is what's really important about using organoids, is that the organoids recapitulate, that is, reform or model the structure of the particular organ we're interested in. And we can go a step further and look at how these cortex organoids develop and compare that to normal brain development. And in a similar fashion to the normal brain, where different cell layers during development get deposited in an inside-out fashion, we also see a similar thing occurring in these brain organoids. And we can see here these key markers of each of these layers being expressed during the culture process of these organoids. So these mouse organoids recapitulate the whole of the formation of the cortex, or corticogenesis as it's called, and we get different expression of different layers at different times during the growth of these organoids, suggesting that it very closely models what happens in the human or mouse brain. And here we have various immunofluorescent images of these organoids showing the expression of proteins that identify different layers of the cortex. And this shows that the formation of all these different layers occurs during organoid formation. So it's a very, very good analogue for some of the major structures in the human brain. It also shows an apical basal axis. So we have the apical region of the cortex in the middle of the organoids and the basal region of the cortex in the external region, or the outside of the organoid. So we have a structure that closely mimics the brain. How can we use this in human disease? And this is not the virus we're all worried about right now, but a few years ago the Zika virus was endemic in Brazil and was causing, or seemed to be associated with microcephaly. So here are the symptoms of the Zika virus infection. It's spread through mosquito bites, and the major symptoms were a headache, conjunctivitis, that's having bloodshot eyes, skin rash, there was joint pain, and a fever. But there was also an anecdotal association. So doctors in maternity wards were showing that there was an association between someone having Zika virus in pregnancy and microcephaly. Was this a causal relationship, or was this just a spurious correlation? Well, we could use these brain organoids to investigate this. It's very difficult to do experiments where you would infect a pregnant woman with a virus and then see if their baby had microcephaly. So this is what microcephaly is. Microcephaly is just a reduction in the size of the head of the baby. And so we can see here, this is a baby with typical head size, and then this is not very severe microcephaly and very severe microcephaly, where we're seeing progressive reduction in the size of the head. So how can we test this in a human model? Mouse brains are different and not great models for human brain development, particularly the cortex, where most of the differences happen in microcephaly. And as I said earlier, we can't simply infect mothers-to-be with viruses and then see what happens to their babies. This is a great platform for a great problem to address with organoids. And what's really nice is we can see here that our brain organoids on this heat map were being compared to other tissues, and we can see here that the brain organoid was very, very similar in gene expression. So we see this red in this heat map, very similar in gene expression to the brain. So our organoids are very similar to brain tissue. Maybe they can be infected by Zika and show an effect. And as I said here, the fetal brain gene expression pattern most highly correlated with the organoids that were being formed in the dish. And sure enough, when we infect, or when they infected their organoids with Zika virus, we saw very obvious changes to the organoids. Not only were the Zika organoids smaller, so they had a smaller organoid area overall. The ventricular zone, which is where all the cortex tissue is, was much smaller in Zika infected organoids. The thickness of the ventricular zone was much less in the organoids, and the area of the lumen, that hole in the middle of the organoids, was much larger. So Zika infection was clearly affecting developing brain organoids, and therefore we could assume or postulate that that Zika could infect a human brain and cause these problems during development of a baby. You could see by immunofluorescence that the organoid architecture was altered. So that lovely organization of the cortex layers that we see in our mock infection is absent in our Zika infection. So Zika infects the forebrain and then alters tissue architecture. And this is what causes the microcephaly in developing babies. So we can use this model that we have of Zika infection in organoids to test drugs. So let's go on to another modelling system. We can model other tissues, not just the brain. We can model the stomach, and we can make what are called gastric organoids. And when we compare these to normal stomach, so the normal stomach structure is here, and we can see this expression of beta-catenin and this nuclear expression of a stomach determining factor PDX1. We can grow gastric organoids from either embryonic stem cells or from induced pluripotent stem cells, and they recapitulate, that is, they model that structure that we see in the normal stomach, that surface expression of beta-catenin and the expression in the nucleus of PDX1. So we can also model the stomach with iPS cells. Why is this important? Well, there's a very well-known bacterium that infects the stomach called Helicobacter pylori, and this is thought to be responsible for some cases of stomach cancer. And so, can we infect Helicobacter pylori with can we infect our organoids with this bacterium? And they could show quite clearly that different strains of Helicobacter pylori could infect the stomach organoids more or less effectively. So here we can see that infection with this G27 causes activation of the c-Met oncogene, and this you can see here a Western blot, and we're pulling down with the c-Met. We can show that we're pulling down c-Met here. Here's the band, and then this shows that it's tyrosine phosphorylated c-Met, so therefore it's active c-Met, it's signaling. And consistent with G27 signaling through Met, we get this increased proliferation in these PDX positive cells. So this G27 strain can cause or predispose to stomach cancer because it activates this c-Met growth factor receptor and induces excess proliferation in the stomach organoids. So let's look at the adult intestine as another example. Here in the intestine, there is again a physical distribution of multipotent progenitors, transit amplifying cells, and differentiated cell types. But unlike the developing central nervous system where we saw layers of cells, there's no separation of the progenitor lineages. But we do have the concept of a stem cell niche where stem cells reside, and then they differentiate into transit amplifying cells or progenitor cells. We heard about this in our adult stem cell lecture in the last half of our lectures. And then these can differentiate after dividing more into our differentiated cells, the gut epithelial cells, tuft cells, and goblet cells. So this is quite a complicated system wherein we have multiple cell types residing within a simple epithelium. Now, can we recapitulate, can we reform that kind of architecture or structure in an organoid? Well, first of all, people experimented with seeing whether they could recapitulate the formation of a complete small intestine just using the crypts, so where the stem cells came from, and growing those in culture. If you took a single crypt of small intestine and grew it in a defined condition in 3D culture, they grew and branched and they formed new crypts. So here we can see new crypts being formed. That's these structures here are new crypts which contain more stem cells. And these started forming within 2 days of culturing these crypts in 3D culture. After 3 weeks, we got these very complicated structures, or they got these very complicated structures, which these sort of protuberances, these things sticking out, are actually the crypts. And we can tell this because a marker of the crypts, LGR5, that's being expressed by these transgenic mice, can be seen in these crypt domains. And if we took a cross section through these organoids, we would see a large lumen on the inside, and the villus domain, so the bits that the villi normally stick into the lumen, are these flat regions here. So although the structure is a little bit different, these organoids clearly mirror the in vivo organization of the small intestine. And what's more is that these individuals then took a single LGR5 positive stem cell, so a stem cell that would have been sitting in the crypts originally, and from that single stem cell can generate an entire small intestine organoid, or a mini gut. Importantly here, all the structures are being made. So we have regions, we have niches in these organoids where cell division occurs and where the stem cells reside. So in these protuberances, which represent the crypts, this is where the LGR5 positive cells reside. We can see this with this green stain here. This is a GFP labeling the crypts, and some of these cells in the crypts are LGR5 negative, and that's these are non-stem cells. These are the niche cells that support these stem cells. And consistent with the stem cells being in the crypts and the transit amplifying cells being just above the crypts, here it's shown very clearly. We can see that in this region here we see active cell division. So these cells are spiked with a metabolic label, EDU, and looked at after 1 hour. And anywhere where we see red indicates cell division. So only in the crypts, or just above the crypts, do we see cells undergoing cell division. So from a single stem cell, we can recapitulate the complete architecture of the small intestine. The same is also possible with pancreatic tissue. Now, pancreatic cancer is very dangerous, very hard to detect, and kills very, very quickly typically. So ways of modelling the pancreas are very valuable in understanding how pancreatic cancer could occur. So you can take the ductal regions of a pancreas, cut these up and grow them in 3D culture. And then you get a structure with a large lumen, these small buds and an epithelium. And we can see this in these micrographs, these microscope images on the bottom. So we get a single duct, and then this single duct starts growing cells around it, and then these globular structures with a lumen form, and we get these budding structures by day 10. So it's recapitulating, reforming a lot of the structures that are seen in normal pancreatic tissue. And if we leave these for a very long time, so 3 months, 6 months and 9 months, they persist in culture because they have stem cells associated with them from these ductal regions. So these are very stable organoids and could be used for various analyses. So in pancreatic tumours, what happens is that lumen gets filled with cells that are derived from the duct. How do we know this? Well, we can express KRAS, the mutant form of KRAS, so the KRAS that is found in 50% of all cancers. And we can express this under the control of PDX1. This mouse line misexpresses an oncogenic form of KRAS in the pancreas. And the result here of this is the growth of these neoplastic cells of a tumour within the ducts of the pancreas. And we can also grow organoids, so we can take ductal epithelium as per normal and grow a normal organoid in our control mice. If we took the same ductal region, which is really a pancreatic tumour, the intraepithelial neoplasia, and grow that, within 3 days we get round structures. But these round structures are different and typically filled with cells. And you can see that here in our organoids, our normal organoids, we see a very large lumen surrounded by an epithelium, and this is completely consistent with what we see in the normal tissue in normal humans. In our tumours, and we can even get cells from what are called fine needle aspirations, where you put a needle into the tumour and aspirate, that's push the plunger up and down several times. Those cells are sufficient to form an organoid. So here we have 3 different organoids made from, or 4 different organoids rather, made from different tumours. And we can see here that the lumen gets shrunk and there are cells residing within the lumen. In some cases there isn't even a lumen at all, particularly in this fine needle aspiration tumour. So the organoids are also modelling tumour structures from tumour-derived stem cells, in comparison to in normal human organoids, where we see normal pancreatic structures derived from normal pancreatic stem cells. And here we can see that the organoids, the fine needle organoid, we can actually sequence the DNA and show that it retains the mutations that we see typically in other just biopsies of normal tumours. So our normal humans have no mutations in any of these genes, and then we can see that in our human tumours there's extensive mutation in some but not all of these genes, and then in one of our fine needle aspirations we also see these mutations. So we can use these organoids that we take from cancers from individuals, and it actually accurately models that pancreatic cancer, and that can be extremely valuable for testing out therapeutics. Let's look at colorectal cancer, where this idea of testing therapeutics with organoids has been taken to an extreme and is actually being used in the clinic. How does this work? Well, we take the tumour tissue from a surgical resection of the colon, and then we make normal and tumour organoids from normal regions and tumour regions of the bowel. And then we can perform on both of these, both the normal and the tumour, we can perform DNA sequencing, RNA expression, drug screens, and then compare these. We can compare the sequence to the sequence we see in the original tumour, and by then we can associate what we see in the organoids to what's happening in the real tumour. And we can compare normal and tumour gene expression and sensitivity to drugs, and using this we may be able to determine for that particular patient what drug treatment may work the best. So here we have a whole exome sequencing of all the samples, and we can see here that some genes we've encountered before are very important. APC, SMAD4, beta-catenin. So a lot of the Wnt signaling pathway is mutated in the colorectal cancers. And so now we have a complete comprehensive molecular characterization of these colon cancers, not just through tissue samples, but also through these organoids. And we'll be beginning to be able to associate certain patterns of mutation, certain patterns of gene expression, certain patterns of promoter methylation, and certain patterns of copy number variation to particular sensitivity to particular drugs. And here's an example of that. What we have here are patients on this side here, and our drugs on this side here, and we're showing the amount of death. So the IC50, the concentration of drug that will cause 50% of those cells to die. And you can see here, based on this heat map, that the different patients have very different sensitivities to the range of drugs. So this systematic screen of 83 compounds identified many differences between the organoids, and therefore many differences between the patients and what drugs may or may not work. So for example, let's look at gemcitabine. So that's the bottom one here. Now, if you were patient 18, what drug would you like to take? So let's look at the black line in each of these. So I can't see any fraction of viability reduction here with MK2206. So that patient could take that drug but would have no effect. It wouldn't reduce the size of the tumour. However, if we look at gemcitabine, look at how efficiently it reduces the tumour size in this patient. But if you were patient 11, gemcitabine would have little or no effect. So the next idea, the concepts, are using organoids to determine a personalized drug treatment regime for that particular tumor. So imagine we had a large database of every patient with colorectal cancer and every drug available on the market, and a determination of which drugs were effective for which kind of tumor. What we would have is personalized medicine. We would be able to more rapidly treat patients with the correct drug. And it would save huge amounts of money and would improve the outcome for these individuals with colorectal cancer, where typically, apart from surgical resection, the outcome is pretty poor. So I've gone through this talk in about half an hour. I don't want to keep you too long. We've looked at how to make organs in a dish. We looked at how we can use iPS cells or embryonic stem cells to make these organoids. We looked at how we can model the cortex of the brain and use this to model the effect of Zika virus infection in the developing brain. We looked at the stomach and how we can use stomach organoids to determine the effects of Helicobacter pylori infection and how that could lead to tumorigenesis. We looked at how a single stem cell from the gut can completely recapitulate the whole of the small intestine. And then finally, we looked at pancreatic cancers, organoids, and colorectal cancer organoids, and how we can use that as a platform to test various drugs to determine a personalized regime of treatment for individuals with these tumors. So I'm going to end there. Why don't we come back at quarter before the hour, so that will be 12 minutes, and then we'll start the next lecture. All right, okay, see you soon.